Hello, this is Shaylin Hansen and Luke Post from Centerpoint Urbana High School, and we are presenting plant growth in Martian soil simulant with and without bacterial inoculant. Colonization of Mars is a very real possibility as NASA and SpaceX both prepare for missions to the Red Planet. In order for life to be sustainable, plants high in nutrients need to be grown as both food and oxygen sources. The Martian soil is principally composed of silicate minerals as well as iron oxide and aluminum oxide. Martian soil is known to be low in essential plant nutrients such as NPK, which can be supplied to legumes using rhizobium bacteria. In this study, Martian soil simulant was used to grow 4 different seedlings both with and without rhizobium bacteria to determine if seedling height and color are affected. Preliminary results show that the seedling in the inoculated Martian soil simulant grew taller than the simulated soil alone.
Our hypothesis is that the seeds planted in the Martian soil simulant without the bacterial inoculant will germinate slower and grow less than in the Martian soil simulant with the bacterial inoculant. According to SpaceX founder Elon Musk, a manned mission to Mars has been planned for the year 2026, with chances of it happening as early as 2024. NASA is also preparing for a trip to Mars in the upcoming years and is leading the Mars exploration program, with the Mars rover Perseverance currently collecting data about the red planet. Villanova researchers Edward Gwynn, Scott Engel, and Alicia Eglin found that Martian soil does not support plants that require full sun to grow very well, such as tomatoes, beans, legumes, corn, and many root plants. Dandelions, on the other hand, flourish in the Martian soil simulant and have significant benefits considering that they grow quickly, every part of the plant is edible, and they have high nutritional value. Other plants that thrive in the simulated environment include microgreens, lettuce, arugula, spinach, peas, garlic, kale, and onions. In preparation for colonization, NASA and the Jet Propulsion Lab made a simulated Martian soil. Plants that are high in nutrients are most important to research because growing conditions and water are limited on Mars. As such, high nitrogen plants that are also drought tolerant were selected for this study. Since it is not practical to take soil to Mars, taking soil inoculants may be more cost-effective.
The materials we used include the following: 12 small pots, 8 circular millipore filters, radish seeds, mothbean seeds, clover seeds, dandelion seeds, earth and soil, Martian soil simulant, and bacterial inoculant rhizobium. We obtained 12 small pots with holes in the bottom for drainage. Circular millipore filters were used on the inside so the Martian soil simulant did not fall through. Next, we placed 250 grams of Martian soil in 8 pots. The other 4 pots were filled with earth and soil to assess and compare germination rates. In the 4 earth and soil pots, mothbean, dandelion, and clover seeds that had been soaking in water were planted, as well as radish seeds that were not soaking in water. In 4 of the Martian soil pots, the same planting procedure was used. In the last set of 4 Martian soil pots, the soil simulant was inoculated with rhizobium bacteria, and the same planting procedure was used again. The Martian soil was watered almost daily with about 15 milliliters of water for the first week. The pots were placed in plastic bags and put in a tank with heat and UV lamps. Plant height was measured almost daily and a water log was kept. And this is our watering log.
As you can see, the seeds grew well in the earth soil and can be assumed to be healthy. On the next slide, you can see the plant growth. In the Martian soil pots without bacterial inoculant, the mothbeans exhibited no growth over the entire study. The clover exhibited small amounts of growth near the end. The dandelion exhibited great growth, but unfortunately flattened out due to lack of water. And the radish seeds also exhibited good growth. More images can be seen on the next slides. In the Martian soil pots with the bacterial inoculant, the mothbeans still exhibited no growth. The clover, on the other hand, exhibited amazing growth and stayed healthy throughout our entire study. The dandelions also exhibited growth, but also failed to maintain when the water was limited, as did the radishes. And more images of these pots can be seen on the next slide.
Our results showed that while plant germination and growth in Martian soil simulant is slower than in earthen soil, the Martian soil simulant can support plant germination and growth. Our research also shows that if the Martian soil simulant is inoculated with the bacteria Rhizobium, the plants germinate and grow better. We also discovered that the plants in the Martian soil simulant need to be watered about every 2 days because the Martian soil simulant does not retain water well. And after a weekend without water, the plants were flattened and no longer able to stay upright.
Based on the data, the hypothesis that the seeds planted in the Martian soil simulant without the inoculant will germinate slower and grow less than the Martian soil simulant with the bacterial inoculant is supported. For example, on the third day after planting the seeds in the Martian soil without Rhizobium, the moth beans and clovers showed no growth, and the dandelions and radishes just began showing signs of growth. In the Martian soil simulant with the Rhizobium, the moth beans still showed no growth after 3 days, but the clover and dandelion both exhibited measurable growth, and the radish had large sprouts. In this study, we discovered that while Martian soil simulant is not as nutritious as earthen soil, it can still support growth for certain types of plants. The moth beans, which are a legume, did not exhibit any growth in either form of Martian soil simulant. The clover seeds, on the other hand, showed great growth in the Martian soil simulant with Rhizobium, even when all the other plants struggled after not being watered, when the watering period was extended to 3 days, making it favorable for growth on the water-limited planet. The dandelions also exhibited strong growth in both of the types of Martian soil stimulant, but struggled when the watering period was extended to 3 days. Dandelions could be a favorable option as well since they were able to grow without the bacterial inoculant. When watered at least every 2 days, the radish also grew well, but could not maintain that growth when the watering period was extended. These data provide evidence that the growth of nutrient-dense plants may be possible in the event of colonization of Mars. Possible factors that could have affected the reliability of our study include our inability to reproduce the exact environment of Mars, including the atmospheric composition and the intensity of the sun. The exact amount of water each plant got could have been slightly different as well.
And here are our references. Thank you.